Okay, so we have a quiz Friday. We have lab tomorrow. We have a lab report due on Friday at 11:59 p.m.
This Friday or next Friday?
This Friday. This is the lab where you built your perimeter.
Yeah.
That lab. Let's see. Help. I'm available Monday, Wednesday, 1 to 5 p.m. by appointment, I'll say. But there's been almost no days that where I haven't announced you ahead of time. I haven't been here the whole time in my office anyway. So let me know after class if you want to come see me. And then help sessions or help session for you guys. Help session 8:30 p.m. on Thursdays by Google Meet. Okay. I think that's everything. That seems like too little, but I think that's everything we've got going on right now. Did I miss anything? Okay, great. So we're going to move on from talking about enzymes and proteins today, and we're going to start talking about lipids. All right. And so lipids are a catch-all class. Of molecules. So they're a catch-all class of molecules. So in other words, when you think about catch-all classes, there are several diseases that are catch-all classes. So inflammatory bowel syndrome, I think is, I don't know which one, there are two of these names, but inflammatory bowel syndrome is you've got some intestinal problems and we don't really know what it is, so we're going to put it in this catch-all category, is kind of the way that works. Epilepsy is actually, I mean, when you think about epilepsy, unless you're experienced with it, you may think of grand mal seizures, which are the shaking seizures. But epilepsy is a class that, or a name they give to any disease where you lose consciousness, essentially, regardless of whether you have those seizures or not. So lipids are like those things in that they're a catch-all class. They are hydrophobic molecules that aren't proteins, nucleic acids, or carbohydrates. So let's say amino acids, or we could say proteins, but okay. Nucleotides aren't the same as nucleic acids, but I'm trying to make this as general as possible. All right, and so they serve important roles, but they are much more diverse than these other things. And so when I talk about our major classes of molecules in this particular course, we're talking the first three are monomers, and you can have polymers, right? So you can have amino acid monomers, and they come together to make protein polymers. You can have nucleic acid, or I should say nucleotide monomers, and they come together to make nucleic acids. And then you have carbohydrate monomers that come in to make polysaccharides, and oligosaccharides. Lipids I put in with the same set of classes, and we often talk about monomers, polymers, but they're not going to really fit in here. Okay, we're going to have some places where we're assembling pieces, but you don't have a single monomer unit that can be repeated over and over and over again. Right? There's not really an example of that in lipids. All right, so then I want to give an example of where lipids are important that you may not have thought about. And my example is going to be sperm whales. And so here's a comic that I had seen a number of years ago. And so the deal with sperm whales, here's my sperm whale. Okay, actually the eye's down here, isn't it? Okay, sperm whales are characterized by they have these really big heads, right? They have these oversized heads compared to the rest of the whale. And so what's that about? And these heads, up here in the head, this is filled with, we're going to say fats and waxes. We'll refine these better. So why do these whales evolve to have these heads filled with all these fats and waxes? Anybody have any ideas or anybody know?
Is it insulation?
It is not insulation. That's a good guess. That's a good use of fats and waxes. It's buoyancy. It is buoyancy, right? So what these whales do is these whales go and they hunt like big shrimp, giant shrimp, and things, not shrimp, giant squid and things like that. And they do it at great depth. So I can't remember. I feel like it's like they can go a mile down and they're okay underwater. So they do it at great depth. And as you go down in water, what happens to the density of water? It gets more dense. Okay, so as you go down in water, you can't compress water much, but you can compress it a little bit, right? You can't compress liquids because they're already sitting next to each other, but you can actually compress them a little bit. If you look at water versus temperature, what you'll see is that water gets less and less and less and less dense slightly until you get to be about 4 degrees Celsius, and then it starts freezing and then it expands. Hopefully everybody in the room knows that ice is less dense than water, right? That's why it floats to this. That's why ice is always on top of lakes when it freezes. But so they're gonna swim way down in the ocean. Okay, and so, whoops, we lost our whales. Okay, so sperm whales hunt in the deep ocean, okay, where the water is more dense. What do you think happens to the temperature as you go down in the ocean too? And it gets colder. All right, what happens with these whales is as they go down and the water gets colder, these fats and lipids that are up in the head actually crystallize and contract, and they make the whale more dense to match the density of the water around them. So this is going to help the whale to maintain density equal to the water around it. So that's a pretty cool use of fats and lipids. It's kind of a fringe use, but I still really like it. You could imagine it's an evolutionary advantage because if you're a whale and you're going to swim way down deep in the ocean, you're going to wait for a big giant squid to come by, right? Or a school of smaller squid. You don't want to be busy. You don't want to be using energy doing this all the time to try and stay down there. Okay. Questions? Okay, so now let's move on and we'll talk more about some more conventional uses or more common uses of fats and lipids. So let's talk about roles of lipids. What do you know that lipids are used for? Okay. Okay, so we've got sterols. Okay, and let's actually talk about you said steroid hormones? Mm-hmm. Steroid hormones. Okay, and what steroid hormones are involved in is its long-range signaling. In other words, when hormones are released into your blood, they go all around your whole body and signal kind of everywhere. All right, so that's one way these things get used. What else?
For energy storage and for like warmth.
Okay, so energy storage. And then I'm assuming you're thinking warmth, you mean insulation?
Yeah.
Okay. Because they also get burned for warmth. What else? They can be structural. Where would they be structural? Yeah, cell membrane, right? That's a structure that holds the cell together. Where else? They can be pigments. So sometimes a lot of the beautiful colors you see, one example that gets used maybe in your book, or certainly seen used, is these beautiful blue butterflies. That color, that pigment they get is due to lipids. I'll show you lycopene in a minute, which is a lipid, and that gives the red tomatoes their color. Okay, they can also be used. I'm just going to say signaling, but let's go ahead and say short range signaling. And we'll have a different molecule for that. It's not going to be sterols. You can use as vitamins. All right, so you've got a number of fat-soluble vitamins, and these end up generally being used as enzyme cofactors. Coenzymes, depending on what field you're in. If you're doing medical sort of stuff, like looking at your MCAT or something, probably they're going to be calling them coenzymes. Okay, those are the ones we're going to talk about, and so let's talk about the various types of lipids that do those roles. And you'll have a given type of lipid may have more than one role. Actually, forgot one. Water repellents. All right, so if you think about waxy leaves and you see water that drips off the leaves, or water off a duck's back, you ever hear that? So that's essentially lipids on the surface of the duck, keeping it from getting all soaked. Okay. So let's talk about types of lipids. We're going to start out talking about triacylglycerols. And so when you think about fats and oils, generally natural fats and oils, well, fats would be natural, but oils that are natural, not dug out of the ground, generally we're talking about triacylglycerols there. We talk about waxes. Okay, like beeswax is actually a wax in this category. We can talk about sterols. Okay, we're going to talk about sterols are long-range signal molecules. They're also structural in nature. We could talk about eicosanoids. Okay, these are short-range signaling molecules. Okay, they're involved in all sorts of things. One that you might be interested in is pain. Okay, so when you take your ibuprofen, what it's doing is it's stopping eicosanoids from being made. And then we can talk about membrane lipids. We're going to spend a lot of time on membrane lipids, the various types of lipids you find in membranes. So starting out, well, actually, let's look at lycopene while we're here. I have a picture of lycopene. So this is a pigment. Lycopene is an example of a lipid as a pigment. Why is this molecule have color?
Because it's aromatic.
It's not aromatic.
Conjugated.
It has a lot of conjugation in it, right? Which does not, aromatic is a special thing. where for conjugation, you have to have rings for it. So yeah, we've got a lot of conjugation here over and over and over again. And as you conjugate all those double bonds together, those pi systems are actually they form one giant molecular orbital, and that giant molecular orbital gets more and more and more stable until what we have here is we have something that's red in color, meaning it's absorbing in the greenish, I don't know my color wheel either. It's absorbing across the color wheel, but I think it's green. Okay, this is absorbed green light, and if this got shorter, if this was shorter, would then get more energetic light would absorb. So maybe it would absorb in the UV, and you would be able to go. If you absorb blue light, then it would be in the UV, that sort of stuff. Okay, so let's go ahead and talk about triacylglycerols. Triacylglycerols it's acyl. So we're using this acyl term, which means you don't have, I mean, we could have them with double bonds in them, but acyl is a carbon chain, okay? So triacylglycerols. Triacylglycerols are going to be made up of a glycerol and 3 fatty acids. All right, so we could imagine we'd have 3 fatty acids, and they don't have to be the same. I'm going to draw these a little bit shorter than they are in general animal systems. In general animal systems, your fatty acids are going to be somewhere, I don't know, 16 to 22 in length. Okay, so and they don't have to all be the same. And in fact, some of them, they can have double bonds in them, or they could have more than one double bond in them. Okay, and that's going to react, it's going to come together with, actually I should do it over here, it's going to react with a glycerol molecule. All right, and when these molecules come together, what you do is if I'm going to make a bond between this glycerol and this fatty acid, what reaction is going to happen there? Dehydration, condensation. We're going to eliminate water to do that. Okay, so we're going to go ahead and take this, and we're going to eliminate water, and same thing over here. I'm not going to be able to neatly draw that. We're going to eliminate water there. Okay. Very possibly, I don't know which oxygens we're actually eliminating there. I did draw it like that. I may have that incorrect in terms of which oxygen is leaving with the water. But what that's going to do, we're going to bring all of those together, and so then we can make a triacylglycerol. So I'll draw it over here. 2, 4, 6, 8, 10, 12. All right, and so we're making a bond here, and it doesn't involve a nitrogen now. It's not in the mid, right? That's what we saw before. We've been looking at for a long time. 2, 4, 6, 8. I feel like I did that wrong. Okay, and then we have one of these left. Whoops, can't lose a carbon. There. Okay, so this right here This is our triacylglycerol. And I forgot something. I forgot my waters. Okay. Questions on that? So triacylglycerols have a few different roles. Actually, they really have only one role. Sorry. Triacylglycerols here, they're going to be energy storage. All right, so in other words, these molecules are used, they're used to store energy. Your body has extra energy. What it's going to do is it's going to store that energy as fat. So these are fats and oils. Your body's going to store your energy as fat because these are great energy-rich molecules. Okay, there are two ways to think about that. These molecules can store a lot of energy because they have a lot of oxidation to go. Okay, so one, they're very reduced, so you can get a lot of energy by combining them with oxygen. Okay, they don't contain much oxygen. So a lot of energy can be gained by oxidizing them. Okay, second thing, they're hydrophobic, so they don't really have water associated with them.
You wrote they contain much oxygen. No, they don't.
They don't, thank you. So carbohydrates are hydrophilic. They have all those OHs sticking off of them, and so water comes and associates with them, and it's very difficult to separate that water from them, and your body just doesn't. Okay, so any carbohydrates you're carrying around your body have a bunch of water associated with it, and that adds to the weight of that storage molecule. So fats are the way to go in terms of if you're trying to store stuff and not carry around as little of it as possible. Questions? Okay. Then what I've done here is I drew a couple different types of fatty acids up above. There are three types I want to consider in here. Say three main types of fatty acids. Okay, they're going to be saturated, unsaturated, and trans-unsaturated. Okay, saturated fatty acids have no double bonds. So if we think about a saturated fatty acid, it's going to look something like this. That's 18 long. We're not going to worry about the common names in here. What we're going to use is something called delta notation. All right, and this has 18 carbons in it, so this would be a C18 fatty acid. Unsaturated fatty acids have one or more double bonds. And I'm going to say cis double bonds. And so an example of that would be this. Oops. Make it super long. Okay, so here I have 2, 4, 6, 8, 10, 12, 14, 16, 18, 20, 22, 24 carbons. That would be C24. And then it's going to have one double bond in it. Okay, and then we put a delta because we need to say where that double bond is. And that double bond is, I'm going to go 2, 4, 6, 8, 10, 12, 14, 15, 16 is where it starts. Delta 16. And this is just an easier way of writing this. We're not, like I said, we're not even going to worry about the common names in here. Not going to bother learning any of them. So now if I had two double bonds, so let's say we've got this one. This one is 2, 4, 6, 2, 4, 6, 8, 10, 12, 14, 16, 18. C18, colon 2 for two double bonds. And then we're going to go delta, and we'll talk about where they are. So 2, 4, 6, 7. This is the seventh carbon here. All right, so this is number 7 when I counted. 8, 9, 10, 12, 13, comma 13. This is 13. 14, 15, 16, 17, 18. Okay, so I was counting correctly because I had 18 again.
Are they superscripts?
They are superscript, yes. I don't care a terrible amount, but yes, they're superscript. Okay, questions on those? How you do that with delta notation? All right, both of these types of fats are natural fats here. So I'm going to say present in high amounts in nature. Our third type of fatty acid is not going to be very present in high amounts in nature. It's mostly in terms of where we encounter it, it's a product of the food industry, although they've mostly eliminated it at this point, and that's trans fats. Trans fats are trans, or have one or more trans double bonds. All right, and so generally you find these in terms of these are human-made by generally you're hydrogenating. So what you do is you do a process where you take the double bonds away from fats and you heat them up, and you take the double bonds, you put hydrogens on, you take the double out, you take them off, you put them on, you can take them on and put them off a few times, and you end up getting, because it's a chemical process, straight chemical process, not an enzyme catalyzed process, you end up with some trans fats, or some trans double bonds in there. All right. Why the food industry loves this, or at least did before ever realized how unhealthy it was was because what it does is it makes them resistant to breaking down. The trans fats don't break down, they don't oxidize as easily, they're not broken down by microorganisms as easily, and so your food lasts longer. Okay, and it still also has, they taste the same, they have a similar mouthfeel, that sort of thing. Okay. It turns out, though, that trans fats are unhealthy for you. You've got about 7 times, they're 7 times as bad for you as regular fats in terms of heart disease. So if you're eating for eating trans fats, it increases your chance of heart disease, a substantial amount of trans fats, let's say, about 7 times. I don't remember the exact amounts on that, but I'll show you a reference that might have that. Or I'll show you how to find that sort of thing. Okay, so a trans fat is going to look like this. They're easier to draw, okay, because we don't need to do our cis double bonds. 2, 4, 6, 8, 10, 12, 14. And so this would be a C14. I haven't really seen a consistent, oh, I'm sorry, the colon, not the colon goes after that. C14. Then we're going to go 1. Then I would say delta trans, what is that? 2, 4, 6, 7. Trans 7. Definitely, I would indicate that. I haven't seen actually in terms of this delta notation, mostly nobody's talking about trans fats, so they're really consistent about whether you put this in or not. All right. But I would definitely want to indicate that. Questions on trans fats? Okay. So these are not healthy. Okay. You get a substantially increased risk of heart disease. And that's well demonstrated. And the thought behind this is that you just don't have the enzymes to break these down. Okay? I mean, you have enzymes to break them down somewhat. If you eat butter, you get it from that, and that's not a problem with, that's just coming out of the cow that way. Okay? So you get some of these trans fats in nature, but not a lot. Your body can break these things down, but you're not really kind of loaded up with the enzymes to do so. So these fatty acids and the triacylglycerols are attached to stick around in your blood more and are more likely to stick to the sides of your blood vessels and cause plaques and things like that. In second semester, we'll talk more about those plaques. Questions? All right, so you may have also heard that saturated fats are not good for you, right? And I'm also going to say, I'm also going to agree with this year. I'm going to say a little bad. As opposed to the trans fats that are really bad for you. Okay? So how do you do this sort of thing? You've got something like this. It's out in the literature. People say that saturated fats are bad for you. And you should have lots of unsaturated fats. They say if you drink gallons of olive oil, you'll be lots healthier. There's all sorts of things like that that are out there. How do you know what to do with this? And as scientists, we know how to look at data. And so what you want to look at for the stuff like that, and we're going to look at saturated fats and polyunsaturated fats, or saturated fats versus unsaturated fats here. When you want to solve or try and make a decision on some sort of dietary or life issue, it's really useful to there's lots of medical studies out there. It's useful to go and look at meta-analyses. Okay, what's a meta-analysis? Meta-analysis is a study of studies. So I'm going to say it's a statistical study of studies. It's not a book report. All right, so that's great. You can get that, but you can do better than that. What's the best kind of study to look at? Anybody know? What was that? No? Okay. Remember, my hearing's not good, so.
Like over time, measuring.
You want to look at something over as long a time as possible. That's good, okay? What you want is, you guys ever heard of a randomized control trial? Okay. So you specifically want a meta-analysis of randomized control trials. All right. So what's the deal with a randomized control trial? Why is that so good? How does a randomized control trial work? Yeah.
It allows you to generalize, and it also allows you to conclude causation.
Okay, the link with causation is the really important thing here. So there are randomized control trials. There are also prospective studies. There's things like the really huge studies, like the nurses study that's maybe out of Harvard nurses study, where they followed these nurses for like 50 years or more. They've not all, you know, they followed nurses for probably, it may be more than 50 years. They've just been following people and having nurses fill out questionnaires on things. But that's a prospective study. That's nurses saying what they did, and then you look for correlations, So let's take an example of, what randomized control studies do is they control for confounding factors. All right, so a good example of this is there was um, a strong belief for a while there, so let's say 10 years ago, that if women going into menopause or getting close to menopause started taking estrogen, that this helped with heart disease. All right, and they had like 400,000 women worth of studies showing this was the case. They looked at women that took estrogen compared to women who don't take estrogen, and they saw a much higher or a much healthier rate, much lower rate, let's say, for the women taking a statistically lower, at least, rate of heart disease in the women taking estrogen. Okay, what could other possibilities be for the lower heart disease rate besides the fact they're taking estrogen? Let's go. It's via.
More concerned about their health.
Okay, they're more concerned about their health. If you're going to go out and you're going to take estrogen, you're thinking about things, right? You're listening to what's supposed to be healthy, and that more than somebody who might be it's life. Diet and lifestyle, okay, which could be related to being more concerned to your health. What else? What else could lead to a different diet and lifestyle? Yeah. Is the estrogen expensive? Yeah, it probably is. Right. So it might be on your health plan. Okay. So it's possibly expensive. It's also, if you are lower income and working two jobs, you might have a hard time going out to get the estrogen or see your doctor to do that, right? Particularly if you don't have great insurance. All right. So there are confounding factors that go together here. All right. There are things that could also lead to a person being more healthy, other than the estrogen, other than taking estrogen. And it turned out when they went and did randomized control trials, all these things were prospective trials. This is one of the examples of a really great reversal there. Estrogen actually either caused a higher rate of heart disease, or it didn't matter, but it did cause a higher rate of death. Okay, so when you look at these sorts of studies, and we're about to look at a study for this, you can see improvements in certain areas. But one of the things you always want to look for is what's the overall mortality in these things, right? Regardless of whether it says it has some effect that you'd expect would make you healthier. Do people live longer, right? Okay, so a bunch of things to worry about here. So randomized control studies control for confounding factors, because what they do is they take a population of people and they randomly divide them into control and test groups. Okay. So they do this by taking a population of people and randomly dividing them into control and test groups. All right, does that make sense? So we just pick, our control group could be all college students. I would just take half of you, and it wouldn't be that I would take half of you and go this half and that half, and randomly assign it. Okay? Because there might be some reason that half of you sat on this side of the room and half of you sat on that side of the room. There's certainly probably social group things happening where you sit, right? You sit next to people you know. But regardless of how we would do that, if we randomly assign a group and it's a large enough group, those groups are the same, and the only difference is whether we treat or not. So you would do estrogen and no estrogen. Okay, and that's how they tease this out. Right? It's not like any prospective studies are good, and they're good for telling you what might be the case. And I actually have a line, I call it like a 50% line for those things. You need to show me, if you're going to give me prospective studies, you need to show me a 50% improvement for me to believe the prospective study. Otherwise, I'm just going to do whatever I want with my life, and I'm not going to listen to your information. That's really how I make these decisions. Okay, so this is the sort of stuff you want to be looking at, and it turns out that there is a group called Cochrane, named after the physician who set it up or started doing this stuff, that really focuses on high quality studies, or doesn't focus necessarily on high quality studies. They take what they've got, but then they rank quality, but they're only looking at randomized control trials. So if you can go to the Cochrane website and you can find a meta-analysis, then that's really useful. All right, and I'm not going to go into, I've taken a lot of time on this. I'm going to show us an example, and over time, I've watched this evolve over the last 10 years. They've gotten to where they'll give a really good layperson summary, okay, and then you can actually get a like medical summary beyond that if you want, and then you could also read their whole study. I've never read their whole study, okay, but I do like their medical summary. This one here, so this is on, I'll take your cookies. All right, so the effect on cutting down of saturated fat on heart disease. All right, so this is a meta-analysis where what they've done is and they have a study here. We wanted to find out the effects of health of cutting down on saturated fat in our food. Okay, so there's some background. We kind of talked about this. Study characteristics. Okay, and so they're assessing the effect, but we only looked at studies of adults 18 years or older. They included men and women. We did not include studies of ill people or pregnant breastfeeding women. Okay, so we're also excluding some studies that might bias our results one way or the other. So what we're looking at healthy individuals in terms of what they've done here. They found 15 studies with 5,600 participants that fit their criteria, meaning that they were randomized control trials at least. Okay, they're going to also they have some other criteria, and you could read what they are. The review found that cutting down on saturated fat led to a 17% reduction in the risk of cardiovascular disease. All right, if we read the medical thing, we'll actually be able to get the error bars on that, which is also useful. But also important for thinking about it. So we've got these benefits. Where's the death thing? It's right in here somewhere. Where was that?
Right after cardiovascular disease.
Okay. Oh, but had little, I stopped at the comma, but had little effect on the risk of dying. So no effect, right? So in other words, eating like this is going to make you less likely to have a heart attack or a stroke, okay? But it's not really likely to increase your lifespan. Right, so you can decide how you feel about that. What I liked as well is this thing they'll do. They'll talk about risk ratios, and so in terms of this summary, they say this means that if 56 people without cardiovascular disease, or 53 people already have cardiovascular disease, I guess, reduce their saturated fat, one person will avoid a stroke or heart attack. Okay, so you can weigh that against, in terms of making decisions on saturated fat versus unsaturated fats. And why I said this year, this has actually been the case for a couple of years, right? This is 2000, I don't know when they published this, but this stuff was going back and forth. They had a report on this. They keep updating these reports, and this is going back for it matters, it doesn't matter. It matters, it doesn't matter. I've been watching this for about 15 years, and it's been on that matters, doesn't matter line for a while. Okay, so you can make a decision about this, and from this I say, well, eating saturated fats isn't all that bad. At the same time, after my fall, I kind of don't want to have a stroke. If something if you tell me it's a 2% risk of killing me, okay, it doesn't bother me all that much. But living with a stroke does. So and it's not, the risk wasn't of killing me, right? So it gives me the information I can make. It gives me information I can use here. Not that bad. Might want to try to eat not unsaturated fats if I can manage. If it's easy. That's me. You'll make your own decisions. Questions? Okay. So you might want to write this down, Cochrane down. This is useful for all sorts of things. If you have breast cancer, you know somebody with breast cancer, you've got a range of treatments that you might have to consider, right? And there might be a Cochrane report or a good meta-analysis out there, randomized control trials, that might inform those decisions, okay? All sorts of other examples of that, though. Questions? Okay. And another really great thing about this is you go to Cochrane, if they have a report on it, and you read that much and you're done. And it's better than whatever novel somebody has written, or whatever several novels somebody has written about saturated and unsaturated fats, and you don't even know. It's just persuasive stuff, somebody doing persuasive writing, right? So really fast answer. Assuming they have a report. Often the answer is we're not sure, right? And if the answer is we're not sure, it needs to be studied more, in my opinion, what I take from that is it doesn't matter, right? So one thing to think about is all the things that really, really matter have already been found out, right? You can have these great debates about stuff, but if you don't take vitamin C, you don't get a vitamin C, you're going to get scurvy, and it's really bad, okay? There's a whole bunch of bad things. Trans fats, okay? Trans fats are pretty bad, and they've got that, okay? Smoking, they never did a randomized control on smoking, but the chance of getting lung cancer is 40 times if you smoke as opposed to not smoking. And that's so big, that's way outside my 50% range, right? That you can go with the prospective studies on that one. So the really bad stuff, the really, really concrete decisions out there have already been discovered. And where people are arguing about stuff, it maybe matters some, it maybe doesn't. Okay, any questions? There's sort of me on a soapbox there, but I'm hoping I've given you important tool here, or a useful tool here for some life decisions you might have to make at some point. Okay. So let's talk a little bit about the melting points of these things. We're going to go back and talk about, we were talking about fatty acids, right? And we were talking about the properties of them. And what I want to do is I want to say that adding double bonds and let's say specifically cis double bonds, has a huge effect on the melting points of fatty acids. Right? It's going to affect how much they stick together. And this may actually be related to this not very large difference you see in terms of fatty acids and heart disease, right? So if we think about this, if we have a bunch of saturated fatty acids, what they can do is they can all line up next to each other and stick really well. We've got these so they can pack in, and we have lots and lots of van der Waals interactions all the way down here. Lots of places we have contact, and that really makes these things have a much higher melting point. All right, so when we think about fats, you get some fat that you're going to use for something. If you're looking at animal fat, let's say, animal fat is more saturated fatty acids than plant fat. You think about plant fat, that's your oils you buy, right? You've got olive oil, you've got canola oil, you've got various types of oils you'll buy, and they have more of these double bonds in them. Now, they are triacylglycerols, but triacylglycerols pack as well. It's just a more complicated picture, right? Imagine you've got 3 of these attached to something. 3 of these attached to a glycerol. Questions?
So more cis bonds means
More cis bonds means
higher melting.
more cis bonds, or more cis double bonds means a lower melting point. And so these things would be a liquid at room temperature as opposed to a solid.
Wait, how? Because if they're closer together, wouldn't that make it harder for them apart?
For these? So these are saturated fatty acids here, right? So we said, let me make sure I wrote it right. Did I write it correctly?
Yeah, so those
Okay.
are solid animal fats.
These are solid animal fats, and these are like plant fats, but we call them oils. Real quick, let me show you some numbers for this. Okay, the numbers are really striking. We're almost out of time, but let's go ahead and look at this. So if I've got an 18 carbon fatty acid, my melting point is 67 degrees Celsius, right? Okay, that's where this thing starts melting. Whereas if I add a double bond, now this thing is melted and it's an oil, right? It's melted, it's an oil at below freezing, okay? And if I add another double bond, it goes down some more. So you see that adding the single double bond makes a huge difference. You can see it here. Let's see, 16, did we get 16? 59 down to below melting again, okay? It's a big difference there. And that's important for membranes and membrane fluidity. Okay, you guys have a good day. Let me know if you'd like to stop